It is often very necessary to culture microorganisms so that we can better study them. If we're able to grow a culture of them, then we can figure out exactly what their nutrient requirements are, what their growth requirements are, and this hopefully will help us in the end to determine how to treat harmful microbes, and then also how to utilize some microbes that could have human benefits to them. In order to culture them, an inoculum is going to be placed into some type of growth medium. The inoculum that we're talking about here is just a small bit of microbes, so a small sample of microbes, and so this will be placed into either a solid medium or it could be placed into a liquid medium. Now, when we do this, the goal is that we will be able to look at individual microbes and see a lot of their characteristics. You can see some of the characteristics listed here on this slide. By determining the characteristics of microbes, we can better classify them and identify them. On this Petri plate right here, you can see how the different colonies, the different spots that we see, they do have different characteristics, and all of these would be things that are useful for classifying microbes. Now, the reason for this whole looking at them, studying the shapes and the edges of the colonies is because if we do this, this is going to help a lot with diagnoses and also with treatment. And it's really all about diagnoses and treatment. Clinical specimens are going to be specimens that are taken from a particular subject. This could be a human subject. This could be an animal subject. You can see in this table right here some of the different places where clinical specimens can be gathered from. It could be a saliva sample, it could be a blood sample, it could be a sample that we get with a needle aspiration from a particular organ. Okay, so if we're able to obtain that clinical specimen, what we want to do with that is we want to then be able to obtain a pure culture. So to obtain a pure culture, what we're saying is that we want a culture that is only made up of that particular microbe. This is what we call an xenic culture. That is a culture that comes from a single progenitor. That means one initial cell started that particular colony. And so a progenitor is referred to as a colony forming unit, or we abbreviate that as a CFU. We have two different methods that are commonly used to obtain a pure culture. This is the streak plate method and then also the pour plate method. If we look at the streak plate method first, the streak plate method is going to use a sterile loop. So this is just a plastic or a metal instrument that is basically dipped into some type of culture that is going to obtain an inoculum from there. And then what we're going to do is we're going to take that and we're going to carefully smear it on the surface of a Petri plate. And the idea is that initially when we touch it down, we're going to have a lot of inoculum on that particular loop. But as we continue to spread it around, we're going to have less and less there. And hopefully we will end up with some places where we get just a single cell spread, and that single cell will then grow into a single colony. So that would be one of those CFUs that we're interested in. If you look over here, these colonies that are circled, those would be CFUs. Those would be considered to be pure colonies, and those could be then taken and studied more carefully. The second method is the pour plate method. And in the pour plate method, the goal is the same. We want to get individual CFUs. But in this case, rather than spreading the inoculum on the surface of this solid media, instead we're going to add it to the media before it's solid. So we add it to the liquid media and mix it up. Then we're going to pour the Petri plates, let them solidify, incubate them for a while so that the colonies can grow. And then what we find is that we will have colonies growing over the surface of the media, and we'll also have colonies growing throughout. In this case, you're getting a better spreading of the inoculum in the media, and we'll probably have more CFUs later on. All right, so both of these methods, the common methods, they work really well for bacteria, and they also work well for some fungi. With both of these methods, though, we're talking about solid media. When we're talking about motile organisms, protozoa and then also some motile algae, these are not going to work well with solid media. They need to have liquid media in order for growth. So for those, we're going to have to approach it a little bit differently in order to obtain a pure culture. If we look more closely at the culture media, nutrient broth is a common growth media that contains powdered beef extract and then also peptones, and that's going to be dissolved in water. Peptones are going to be amino acids or small polypeptides, and so this substance is going to provide a lot of the nutrients that are needed for that particular microbe, and then we may have additional things that are added into that media depending on the requirements of the microbes that we're trying to grow. This media can be made in a solid form or it can be made in a liquid form. If it's liquid, we don't have to add any type of gelling agent, but if it is a solid form that we're trying to make, we do have to add agar. Agar is a really good thing to add to the media because, first of all, it does provide a nice solid surface that is going to support colony growth. Another thing is that most microbes cannot dissolve it. They can't break it down. That is good because if it was something that was going to be broken down by microbes, initially we'd have a solid surface, but as the microbes grew, they would dissolve this surface stability as it broke down. So agar is not broken down by most microbes, which means the plates do last for a while. It's also something that's very easy to dissolve. So it is going to be something that's easy to prepare. And another thing is that solid agar does not melt at really low temperatures. So it doesn't melt at anything below 100 degrees, which means that we can still take our plates and place them at a warm growth temperature, human body temperature, or even at temperatures that are warmer than that, and we can expect that the media is still going to hold its form. So this is just a look at how the plates would be poured or how we can pour different tubes of media. Basically, the nutrient broth with the agar is melted and it's sterilized, and then as it's still a liquid, the plates will be poured, they will be set to the side, and then they will be allowed to come to room temperature, which is going to cause the plates to gel up. If we're talking about tubes, the tubes can have liquid media in them, obviously. We can have just a solid media in the tube. Or if you look at the tube that's in the middle, that one is what we call a slant. And with a slant, basically the tubes, the media is poured into them when it's still liquid, and then the tube is placed at an angle while it solidifies. And what this does is it allows us to basically put the microbe maybe up on the top, and then if there's any liquid, it can kind of drain off of that to the bottom so that we do keep our microbe out of any liquid that might be accumulating in the tube. There are different types of culture media that are used. We separate those into different groups. The first one is what we call a defined media. With defined media, we know exactly what is in that media, exactly what's there and how much of it is there. This is just an example one listed here. You can see very specifically this media has this many grams of glucose, this many grams of calcium chloride, the whole way down. Most of the time we are not growing microbes in a defined media. Usually we grow them in more of a generic media, and we call this a complex media. We don't know exactly how much of every specific chemical is there, but we know that it is providing for the needs of that particular microbe or for a wide range of microbes. With these, they are usually taking nutrients that are released by a partial breakdown, a partial digestion of yeast, beef, or soy proteins. So with these, again, the exact composition is not known, but it is providing kind of a good generic substance that we can grow a lot of different microbes on. A selective media is going to be a media that contains substances that either favor or inhibit the growth of certain microorganisms. So here in this picture, we can see an example of two different selective medias. One is favoring the growth of bacteria. One is favoring the growth of fungi. With selective media, when we do vary the chemicals that are there, that is going to alter the pH, which in some cases is enough to select or inhibit specific ones. We can also leave out a particular chemical that something needs to grow, and in that way, by just omitting that particular chemical, we're ensuring that that microbe is not going to grow on this particular substance. In some cases with our selective media, we are trying to increase the abundance of some cultures or some microbes that are a lot less abundant. So that is a type of selective media because we're trying to favor the growth of this rare or uncommon microbe. With differential media, we have a media that is going to produce different visual characteristics depending on the organisms that are growing there. There's two examples listed here. The Petri plate is an example of blood agar. Blood agar plates give us different visual results depending on how the microbes are able to metabolize hemoglobin. So you can see there's 3 different ways that they can appear on these plates depending on their metabolism. The second picture here is showing us a difference that we can get based on pH changes in there. So we can have, in our differential media, we can have different pH indicators that have been added, and they will help us to detect differences in carbon dioxide production or production of other acids. Anaerobic media is going to be called reducing media. And that's because this media is going to contain compounds that chemically combine with free oxygen. And when they do that, they will be removing that oxygen from the media. So they are removing it completely from where the microbes might be growing. This is important if we are talking about some of those obligate anaerobes, which are the ones that cannot grow in the presence of oxygen. For these types of Petri plates, many times they're also placed into a special chamber that is also going to help keep the oxygen out so that it's not interfering with the culturing of the microbes. Lastly, we have what are called transport media. Transport media is media that is basically put there just to help us transport the microbes from point A to point B. So this is going to be used to carry or transport clinical specimens from the site of collection to the place where they are ultimately going to be cultured so that we can obtain that pure culture and do our diagnoses or actually study them so that we can come up with treatment methods. Some organisms require very specialized culture conditions, and this means that we have to come up with special methods in order to culture them. Many microbes have to be cultured in living cells. They're not able to survive on just a petri dish surface or in liquid media. One example of one that has to be cultured in living cells is going to be the microbe that causes leprosy. For that particular one, it is commonly cultured in armadillos. Other examples of microbes that have to be cultured in living cells are viruses. Viruses require the use of host machinery in order to replicate, so they would never grow on a petri dish surface or just in a liquid media if you don't have a living cell to replicate for them. We also have some microbes that require very specific carbon dioxide levels or oxygen levels. With these, typically those particular concentration levels have to be created using specific chambers. If we talk about storing culture, we do need to have methods to store cultures both short-term and then also long-term. Refrigeration is a really good way to store cultures short-term. With refrigeration, a typical refrigerator is at about 4 degrees Celsius, and that 4 degrees Celsius is going to allow us to store many petri dishes or liquid cultures maybe for several weeks, maybe even up for a month or 2. Deep freezing is another possibility. With deep freezing, we're talking about getting those cultures in a freezer that can maintain maybe somewhere around -70, -80 degrees Celsius. With these cultures, they can last years. They can last maybe even decades if they're deep frozen. And then lyophilization is kind of a freeze drying. So in that case, the cultures are dried out and they're frozen. And in that case, we're going to get the best long-term storage of our culture. So those are going to definitely last decades if we do store them by that method.